single cell analysis. And I would like to spend a few words on an introduction, the way we approach this topic, and then we will zoom in to single cell genomics, which is currently the biggest segment within the single cell analysis. Single cell analysis as a term is basically translated best as being the study of genomics, transcriptomics, proteomics, and metabolomics at the single cell level. And this is nicely shown in the right picture, where basically you see a sketch of a cell taking the cell core where the DNA is basically stored. The DNA translates and transcripts to the RNA, which then brings the code outside towards the proteins, and the proteins are being synthesized. And all these segments can be analyzed separately through using single cell analysis tools. As I mentioned, probably the biggest segment within the single cell analysis arena is single cell genomics. And typically, if you look at things like liquid biopsies, which I'll come back to in the next slide, you'll understand why. So here we really look at the DNA on a single cell level. And to be able to study these single cells, we need sorting methods, sorting technologies to separate those cells and isolate them from an organism's tissue according to their type or geometry or size. And usually these are, as I mentioned, we rely on differences in morphology, but also surface protein expression, for example. And the resulting populations of cells have very important applications in research and as therapeutics. And that's why this area, I believe, is very rapidly growing and also very much connected to the fields of next-gen sequencing that I just discussed. We look at the workflow steps for single-cell genomics. Basically, we can divide them in 3 steps. The first being, let's say, the preparation or the biopsy. And this can be a normal biopsy, like a cell tissue, which is regularly still being done today, for example, in oncology. But it can also be a liquid biopsy, where basically blood is extracted to search for circulating tumor cells, CTCs. The second step, and I'll address that a bit more in detail, is how can you prepare then this sample and how can you isolate the cells of interest? So which cells do you really want to look at and how can I get them out of this biopsy or tissue? For that, you can use technologies like droplet generation and cell sorting. And last but not least, of course, is the analysis itself. Once you have been able to isolate the cells of interest, you want to analyze them very accurately on a single cell level. And as mentioned, you can use techniques like NGS or PCR-based techniques, which are also commonly used in the genomics applications. The right picture, as you can see, shows how you can capture a cell by using droplet technology, where reagents and primers are being brought together with the cell. They flow through a first microfluidic channel, then oil is being entering from the side, which is creating a dispersive water phase or aqueous phase, capturing each individual cell together with its primers and reagents and being able to do analysis in a single droplet, which contains a single cell. Some platform examples. Again, this list is not comprehensive, but it shows some of the market players out there which offer these kind of platforms to analyze and to isolate cells. Berkeley Lights has a very nice platform to manipulate cells on a singular level. Sphere Fluidics, Fluidigm, Bio-Rad, and there's a number of more companies that I'll show in the next slide that offer these kind of single cell analysis tools. And again, looking a bit into the market, you see that there's multiple players out there, and these days there's still a lot coming in new. For example, 10x Genomics, that is dealing with single cell genomic applications, not mentioned in this slide, but certainly an important player in this field, amongst the others that you see in this list. And all these companies mentioned here are using some kind of microfluidics-based technology to analyze and isolate cells. Looking at the market size, in the past 5 years, similar, I would say, to the NGS market, growth 15 to 20% on average each year, and still growing heavily. But the reason I selected this slide to show it to you today is if you compare the total market in terms of value, being, let's say, in the hundreds of million or in $100 million in 2020, and you compare it to the number of units being processed, you can see that there's really, these parts have quite a high added value in terms of cost. So it's not a cheap consumable that only can cost a few euros. We're really talking about high-end complex devices that add value to the product and that can cost something as well. So a very interesting market for us to be in terms of looking at consumables. Now, looking a bit at the trends, this slide contains quite some info. I will not mention all of the aspects. I'll just go through it and highlight some keywords. Single cell analysis is a disruptive technology, especially looking in fields like neurology, stem cell biology, oncology. And as I mentioned already, amongst this single cell analysis, single cell genomics is the fastest growing approach, in which genomic technologies are being used to look at DNA and RNA and really look at the single cellular level. Also, what we see is that with the advances in genome editing, high throughput systems and microfluidic technologies have enabled the rapid and parallel analysis of smaller samples that are common with single cells. And single cell analysis markets are rapidly emerging in various industries. The field is growing beyond basic research, basically happening the same as what we saw with NGS. I think single cell analysis is now following that closely, and we're looking into applied fields like molecular diagnostics applications. Now, I'd like to zoom into this workflow, and especially the second step. I already mentioned that we try to combine our microfluidic technologies with the workflow of these applications in the market. And here I'd like to zoom in a little bit into the droplets, and then I'll go into further detail regarding cell sorting. I have one slide about droplet generation. In the top picture, you see a single droplet generator, and to the right, you see how the droplets are being created. These are typical, I would say, standard products that we can make in glass, but also in other materials and polymers, and that we offer to our customers. More importantly, we get more and more demands. In the past years, we've seen that trend growing to increase throughput of our droplets. So we designed and developed parallel droplet generators as well, which you can see in the bottom picture. This can go from 4 channels to 8 channels to 16 channels and beyond, depending on the throughput that you need. And you can see in the image in the movie appearing in the top or in the bottom right how this works. Basically, you have individual droplet generators that are all pooling at the end of the chip, creating a higher throughput of droplets. Also important to mention, these droplets can be either with water droplets in a continuous oil phase, or vice versa, oil droplets in a water phase, depending on how you coat your surface. So we can be very flexible with offering these systems, either in a stage where you create water droplets or in a stage where you can create oil droplets, depending on your application, of course. That's all I want to address regarding droplets, because as you know, there will be a Micronet webinar coming up on the June 2nd about droplets, and we will go in much further detail there during that webinar. Then let's look at the sorting technologies that we offer at Micronet, or I should say that we are working with together with our customers and design chips around. The first technology I'd like to address is acoustophoresis, which also known as SAW, surface acoustic waves, is a powerful technology where external acoustic force is applied to a microfluidic channel in order to concentrate cells or to sort cells, if you will. And you can see in the image what is happening, or in the video. In this case, we have a yeast cell concentration where yeast cells are being beamed and focused into the central channel, and thus you can concentrate the cells that you would like to have based on specific needs like size, for example. So this technique is used a lot for cell washing, for example, cell concentration, instead of using filtration techniques. The second technique, which we call microfluidic switching, works in a bit of a different way. As you can see, this is an animation of a microfluidic channel in which cells are lined up individually and pass with a high speed through a laser area. The laser detects whether a cell is of interest or not based on its morphology, or based on its chromosomes, or there can be different properties that are analyzed. But the laser can look through the cells individually very rapidly, a bit like flow cytometry techniques. When the cell is selected for sorting, it will interact with an actuator chamber, as you can see in the mid of this video, and the chamber will generate a very slight pulse to push that cell aside towards the wall of the microfluidic channel, and then it's moved out of the way, basically. So the blue cells in this case are being selected, the black cells are just moving to the waste. And the blue cells are actually the cells of interest that we want to select, for example, for doing single cell analysis, or for isolation for cell therapy, for example. The nice thing about this technique is that the cells are not in contact or are not damaged in any way. They're just slightly pushed aside without having any damage to the cell walls. The third technique that is used by some of our customers is called magnetic levitation. It's a technology that has been out there, but it should not be confused with magnetic beads-based technology. Because in this case, there's no beads involved. It's just an external magnet that is closely wrapped around a microfluidic channel, where cells are being, let's say, magnetically levitated, similar to the Japanese bullet trains that are driven by magnetic force, but are not in contact with the actual railways. So it's really the magnetic field that will make the cells levitate at a certain level. And in this way, you can also nicely distinguish, for example, between circulating tumor cells, white blood cells, red blood cells, platelets, etc. So it's a very nice and new technology to sort blood cells. Fourth technology that is used in the market is known as dye electrophoresis, or DAP abbreviated. There you need electrical forces, an alternating current field, which is offered through finger electrodes, as you can see in this image, on top of a microfluidic channel. And here the nice thing is that you can basically manipulate your particles or your cells by applying a specific electrical field at certain times in your protocol. So if you, for example, want to trap cells to be able to wash them, you can apply a specific electrical field which will hit cells of a certain size or charge, and those cells will be trapped on the finger electrodes until they are washed and can be pre-selected into a specific channel. And in this way you can address also cells based on charge and size in a very selective way. Fifth technique is called pinched flow fractionation. I only have this picture. The left picture is basically zooming into a microfluidic channel. What I can say about this technique, it's similar to, I would say, the microfluidic switching technique, where you basically push cells aside towards the channel wall by using an additional flow. And here you basically make use of the laminar flow that is commonly occurring inside microfluidic channels. And by using that force, you basically also push cells towards a wider channel in a different direction. Bigger cells will tend to go to the center area of the channel, smaller cells will tend to go to the edges, to the wall of the channel. And in this way you can select the cells further down the channel at the end of the chip. Final technique I'd like to present, and I'll go into a bit further detail there, is called deterministic lateral displacement, better known as DLD, where basically you use microposts, as you can see in this image, which are micro-embossed into a polymer device, and you are also selecting cells or sorting cells based on their size. And this is especially powerful when, for example, you want to sort blood cells, and I'll show that in the next slide. This is an example of a customer project we are working on, a product development of a cartridge that is able to sort red blood cells from white blood cells. In the left image, you see the animation, how this technique works. And I try to explain in the words above how it basically, what it comes down to. This cell processing system is developed by GPB Scientific, and it employs a microfluidic chip technology, which is based on DLD processing, and it gently and uniformly separates the blood cells on the basis of size. And you could compare it to a mechanical coin sorter. I think that's the best comparison you can make. Basically, if you look at the animation, a bigger coin is moved towards the right, the smaller coins are moved towards the left. And the fact that this technology is actually, it's working, I tried to show in the video, the real-time video you see in the center. which was sent to us by our customer after testing their first device. This is a multi-layered cartridge, which I'll show in the next slide, where actually red blood cells are being separated from white blood cells. And if you look closely, you can see a whitish channel in the center area of each lane and two reddish channels at the sides, where the red blood cells are pushed to the side by using this coin sorter, and the white blood cells are more moved towards the center area. And by using this technology, the accuracy of selecting cells has increased, and basically it can replace centrifugal technologies that are out there today. This is how the cartridge and the instrument look like currently. As I said, it's being developed, and our customer GPB is going to commercialize this and bring it to the market. The device at the left-hand side is a polymer device consisting of multi-layers that contain all these lanes that have the micropulse for the cell sorting. And I know we have been talking about microfluidics in this webinar, and usually people tend to compare that with volumes like microliters, or maybe even less than that, nanoliters, picoliter droplets. But here really we are talking about increasing fluidic throughput up to levels of 500 ml per hour, which is basically the size of a blood bag that you want to sort within an hour time frame. So quite a powerful technique if you're able to bring all these layers together.